Hello again and welcome to my channel. And this is part of a coronavirus COVID-19 crash course and it's the second lecture. Building a coronavirus. Okay, so we are not going to the lab to actually build a coronavirus. We will leave that for another occasion. Right now we are going to discuss about the composition and structure of coronaviruses because this information is essential to understand how coronaviruses work and it helps us to devise new therapeutic approaches. This is a reminder. I am David Scores. I'm a virologist and immunologist, and this crash course is intended for everyone, and the information that you will find here is faithful and precise. If you are interested in this crash course, you can subscribe to this channel by clicking on the mousing thing that will appear here during the presentation. So, what is the first thing that we need to make a virus? We need the instructions to build it. We need a genome, and this genome consists of a set of instructions that the virus is going to introduce into the cell so that this cell will produce more progeny viruses. So here is the coronavirus genome. It's not DNA, it's an RNA molecule, and it has the characteristic of being the longest RNA genome in nature. Here is represented as a single molecule adopting a kind of helical structure, and it contains all instructions to make a coronavirus. In future videos I will explain how this information is encoded. Once that we have the RNA genome, we need to protect this genome using a nucleocapsid protein. As you see here, the nucleocapsid protein, represented as orange squares, associates with the RNA, and it makes a kind of a spherical structure called the virion core. As you see here, a purified virion core under the electronic microscope is a kind of a spherical structure, and you can even make out the strand of RNA associated to the end protein, like a kind of ball. The core in these conditions is still very fragile. So the virus packages this core within a membranous envelope, like a kind of plastic bag here. And this envelope comes from the cell's endoplasmic reticulum and Golgi. Now that we have the core inside the viral envelope, we need to add further proteins in the membrane, such as the membrane protein here and the envelope protein over there. And these proteins, what they do, they stabilize the structure of the internal core of the coronavirus within the viral envelope. So how does the virus enter the cell? In this condition, the virus cannot enter the cell. It needs something else. It needs a crown of spikes that surrounds the virus. And these spikes or peplomers are made of the S protein, and this S protein binds to receptors present on the surface of the cell to be infected. So coronaviruses use these spikes as a way to get into the cell and infect it. Here we have an electron microscope picture of the SARS coronavirus, and you can see inside the spherical structure over there, surrounded by this characteristic crown of spikes. This is a electron microscope picture of purified coronaviruses. These are big coronaviruses, and here you have the SARS coronavirus picture. So all coronaviruses, they look the same under the electron microscope, and they have this characteristic crown of peplomers there. They are very small, something like 100 and 120 nanometers in diameter, which is 0.000001 meters. So it's impossible to see this virus, this type of viruses under the normal microscope. It's not like in the pictures when you have the main character looking at the standard microscope and then they see the Ebola viruses swimming around and so on. No, you really need electron microscopes in order to visualize these viral particles, which are rather beautiful under the electron microscope. Finally, thank you for your attention, and I hope I will see you again in the next video. Thank you very much.